I'd like to start by thanking Phil for organizing this seminar series. I'm really excited about it, and I'd also like to thank him for including me. I'm a postdoc in Josh Rabinowitz's lab at Princeton University, and today I'm going to talk about my work using quantitative methodology to study metabolism with a focus on glucose metabolism. So my research is interested in chronic metabolic changes that lead to increased risk of disease. And so we know that millions of Americans, over 40%, have obesity, and a large fraction also have metabolic syndrome and non-alcoholic fatty liver disease. And these are chronic changes in metabolism that lead to increased risks of more life-threatening diseases, such as type 2 diabetes, heart disease, and cancer. And so my research is focused on understanding metabolic pathways that underlie these chronic metabolic changes, how these pathways can become dysregulated with a goal of preventing progression to disease. And so my talk today will focus a lot on understanding these underlying metabolic pathways that become dysregulated in chronic metabolic diseases. And so the first half of my talk will focus on glucose metabolism, with the specific question of what tissues use glycolysis to catabolize glucose. And then in the second half of my talk, I will focus on de novo lipogenesis, which I'll talk about a little bit later. So let's start with glucose homeostasis. So to balance glucose homeostasis, you need to balance production and consumption of glucose. So in the fasted state, glucose is produced by gluconeogenesis and glycogen breakdown, and then it can be consumed by these three major routes. One is through catabolism of glucose, so glycolysis and eventual oxidation in the TCA cycle, and storage. So two routes of storage are production of glycogen, which is a polymer of glucose, and then conversion into fat through lipogenesis. And we know that one challenge to maintaining glucose homeostasis is the influx of glucose from food. And the most important regulator of glucose homeostasis is insulin, which can decrease endogenous production of glucose in response to food and increase consumption into tissues and increases all of these glucose consuming pathways. And so today I really want to focus on the catabolic pathways, glucose, glycolysis, and the TCA cycle, and this branch here, oxidation. And that's because this is the main way in which glucose is cleared from the body, while these other two pathways are storage for use of glucose as energy later. So glucose, when it's taken up into tissues, will be phosphorylated and then catabolized via glycolysis, with the end product being pyruvate in the tissue. Then pyruvate can be converted into lactate for export into the circulation, or it can be used in the TCA cycle to make more ATP. What's becoming increasingly appreciated is that most tissue pyruvate actually will be converted into circulating lactate before being taken back up into tissues and used in the TCA cycle. And so when I joined the Rabinowitz lab, I became really interested in this idea because this suggests potentially that glycolysis and the TCA cycle could be uncoupled. Glucose could be taken up by one cell type, converted to pyruvate and secreted as lactate, while another cell can take up lactate and use that lactate in the TCA cycle to make energy. That means that some cells could potentially make energy from carbohydrate without ever doing glycolysis. And so I thought this was a really interesting idea. And so the question that I've asked during a lot of my postdoc work is which tissues actually do glycolysis? And when I first started working on this project, I thought that there must be a lot known about glucose metabolism. So is this really a novel question? And I think that there is obviously, there's decades of work on glucose metabolism, but there's a few reasons that this is not fully understood yet. One is that the workhorse of studying glucose metabolism is 2-deoxyglucose. And this is an analog of glucose that can be taken up into tissues and phosphorylated, but cannot be catabolized downstream in glycolysis. So this is really powerful because we can measure the accumulation of the 2-deoxyglucose phosphorylated version in tissues over time to measure glucose uptake and phosphorylation. However, its disadvantage is it can't be used to study downstream glycolysis. Another reason I think that there's been a lack of literature on glycolysis in vivo is that glycolytic intermediates are actually quite low in concentration. So what you can see here is fructose bisphosphate, a metabolite in mid-glycolysis, is actually quite low in concentration. And here I'm showing it in comparison to succinate, a TCA cycle intermediate, which is 10-fold higher in concentration. And I think this has led to a lot of analytical challenges and a lack of reporting of glycolytic intermediates in the literature. So I think this is a novel question to ask: which tissues do glycolysis? And in order to study this, I am using stable isotope tracing. So stable isotopes are heavier, usually heavier isotopes of a molecule, such as carbon, which usually has a molecular weight of 12, but carbon-13 has an extra neutron and therefore a molecular weight of 13. So you can buy labeled glucose that can be used by cells and tissues just as 12C glucose, and will be catabolized and used in enzymes in downstream metabolic pathways, just like regular glucose, and therefore we can measure the incorporation of these carbon-13 molecules from glucose into other metabolites. We can extract these metabolites, then measure on the mass spec, and when for each carbon-13 that is incorporated into the metabolite, we can see a molecular weight shift of 1. And so we can do this in vivo. We do this by intravenously infusing our carbon-13 tracer. So here I'm showing we infuse the uniformly labeled carbon-13 glucose, and we do this through a jugular vein catheter, so that the mice can freely move around the cage while we're infusing. And we can also implant a carotid artery catheter to draw blood so that we can measure what happens to this glucose without ever having to stress out or touch the mouse. And so what we can see is that the labeled glucose appears in circulation. This is the blood, the labeling in the blood that I'm showing here over time. And because we infuse at a constant rate, eventually this amount labeling will reach steady state. And we generally do these infusions for 2.5 hours. And when we do this with glucose, one of the first things we see is the appearance of labeled lactate in circulation. So lactate is the product of glycolysis. And so we can see that it is becoming labeled over time, and by the end of our infusion, over 50% of lactate is labeled, suggesting that more than 50% of lactate comes from glucose. So it seems that glycolytic flux on the whole body level is very high. So which tissues are actually doing this glycolysis? To do this, we collect tissues at the end of this infusion, and we had one simple idea in looking for glycolytic tissues, and that is that the glycolytic intermediates of glycolytic tissues should label from circulating glucose. And if this tissue is responsible for a lot of lactate production, then we would expect the labeling to be higher than that of circulating lactate. So just briefly, I want to mention how this data will be represented. So when we measure the labeling in a tissue metabolite pool, here I'm showing an example of glycolytic intermediate fructose bisphosphate, where 1 of every 10 molecules is labeled, which would be about 10%. Because we infused our tracer at a tracer enrichment, which is usually about 20% or less, we normalize the labeling of the metabolite to that of the labeling of the traced metabolite to get a value for how much of that metabolite comes from our tracer or from our traced molecule. So here in this example, about 50% of fructose bisphosphate comes from circulating glucose. And if we saw 20% labeling in the tissue, that would be 100%. So I just want to advertise that we have a perspective on in vivo tracing coming out in Nature Metabolism that was spearheaded by another postdoc in the lab, Caroline Bartman, so watch out for that. So which tissues do glycolysis? Which tissues have a lot of labeling in their glycolytic intermediates from glucose? So here's an example. If 100% of glycolytic intermediates come from glucose, which is what we often see in cell culture, you would expect to see 100% or 1 in your normalized labeling. And the amount of labeling we see in circulating lactate is about 55%. But when we look at glycolytic intermediates, and here I'm showing 3 glycolytic intermediates at upper, mid, and lower glycolysis, what we were surprised to see is that the labeling is quite low. And so the first reaction we had to this data was we wanted to make sure this wasn't an artifact. So we did some work making sure our measurements were accurate, and we made some slight improvements. We tried to infuse our tracer for shorter and longer amounts of time to see if this labeling was at steady state, and indeed it is. We can see this amount of labeling with very short infusions, and it does not increase when we increase the infusion length. And we also tried to account for stress to the animal that might cause glycogen breakdown, but nothing that we tried really changed the amount of labeling that we see here. The one thing that did work was we just looked at more tissues. So on the next slide, I'm going to show more tissues where the glycolytic intermediate labeling that I'm showing here is all averaged into one value. So here is the average labeling of glycolytic intermediates from glucose in about 20 tissues. And the gray here is where the labeling of circulating lactate is. So if a bar is in this gray area, that means it's labeling more than circulating lactate. And so we had 2 striking observations from this data. One is that the majority of tissues label about 50% from circulating glucose. And this suggests that the majority of glycolytic intermediates is not coming from circulating glucose, and there must be another source. And I'll talk about that a little bit more later. The second striking observation was that we saw that these muscles are labeling on opposite ends of the spectrum. So on the left, you can see the diaphragm and the soleus are 2 muscles. The diaphragm is important for breathing, and the soleus is a muscle in the leg, label on the highest end of the spectrum. And they label as high as whole blood, which is dominated by red blood cells that don't have mitochondria and rely completely on glycolysis for their energy production. And then on the opposite end of the spectrum, we have the quadricep muscle and the gastrocnemius muscle. And so we were really interested in why these muscles are behaving so differently. And many of you probably know that there are different types of muscles and muscle fibers. So the mouse soleus is made of a lot of slow twitch muscle fibers that are high in mitochondria and high in myoglobin. The mouse diaphragm, which labels very similarly to the soleus, doesn't have as many slow. Oops. Okay, so the mouse diaphragm that has about equal labeling to the mouse soleus has a lot of, does not have a lot of slow twitch fibers, but has a lot of fast twitch fibers that are also high in mitochondria and myoglobin, which are these type 2A fibers. So it seems that the common common thing here between the soleus and the diaphragm is that both of these muscles have a lot of mitochondria and myoglobin. So we came to the conclusion that maybe it's red muscle that is actively using circulating glucose in glycolysis. And in contrast, the quadriceps and the gastrocnemius muscles have mostly type 2B fibers, and these fibers are low in mitochondria and low in myoglobin and label a lot less. So it seems that red and white muscle seem to be different in their glucose use. So red muscles use circulating glucose for glycolysis. So the next question we had was, what is the source of these other glycolytic intermediates in tissues? And one obvious source is glycogen that is stored in the tissue. So in order to look at the contribution of glycogen to tissue glycolysis, we needed a way to trace glycogen. However, because glycogen is a polymer that is stored within tissues, we cannot just infuse labeled glycogen intravenously. So we needed to come up with a strategy to measure the contribution of glycogen to glycolytic intermediates. And so what we came up with was a pulse chase infusion of carbon-13 labeled glucose. So we infused carbon-13 labeled glucose for 16 hours overnight while the mice are eating. And this should lead to labeling of circulating glucose and glycogen synthesis, and therefore the labeling will be incorporated into glycogen in the tissues. Then we stop the infusion in the morning and fast the mice. And during this time, the circulating glucose will lose its labeling, while the glycogen, which has a much slower turnover, should maintain labeling. And when we collect tissues, hydrolyzed glycogen, and look at the labeling, that is exactly what we see. We can see that there is labeling maintained in the glycogen pools in the muscle, the liver, and in other tissues. So this is a good tool to use glycogen as a tracer. And so to demonstrate how we can use this to look at the labeling of glycolytic intermediates, I'm going to focus on the quad. So when we look at the quad and we normalize other metabolites to the labeling we see in quad glycogen, what we see is that circulating glucose and lactate are still labeled about 75%. And what we know from our short-term glucose infusions is that when we infuse and label circulating glucose, we get very little labeling of glycolytic intermediates. And based on that, we can predict how much labeling we should see in glycolytic intermediates in this experiment. Therefore, any labeling that we see above this amount should be from glycogen that is stored within the tissue. And indeed, we do see a lot more labeling than what we would expect from circulating glucose. And in fact, the labeling was even higher than what we observed in glucose, which makes it impossible for this labeling to come solely from circulating glucose. However, we wanted to get a more quantitative estimate of how much of these glycolytic intermediates in tissues are coming from circulating glucose versus stored glycogen. So we used some math that was actually developed by Tony Hui, who was a postdoc in our lab and is now faculty at Harvard. And he thought about a problem that has been known in the tracing world as a secondary tracer problem. And so when you infuse glucose and measure its labeling in glycolytic intermediates, the labeling that you observe will be a sum of the direct contribution from glucose and the indirect contribution of the contribution from glycogen, because glucose will label glycogen, and then glycogen can be broken down to make glycolytic intermediates. And so in this case, we can write an equation where the labeling we observe is the sum of the direct contribution from glucose and the indirect contribution from glycogen. And in this case, we have two unknowns, and we don't have enough information to solve for these unknowns. However, what Tony realized is we can do another experiment in which we can label our other secondary tracer, and that will now become our primary tracer. So in our pulse chase experiment, we labeled glycogen, and therefore the labeling that we observe in glycolytic intermediates is the sum of the direct contribution from glycogen and the indirect contribution from circulating glucose. And so now we have two equations with two unknowns, and we can solve for the direct contributions. And so when we do this, this is what we get. So the most striking tissues are, of course, the quadriceps, which gets almost all of its glycolytic intermediate from glycogen, and the soleus, which is a red muscle, which gets the majority of its glycolytic intermediate from circulating glucose. And then the rest of the tissues are somewhere in between those two. What was striking to us was when we started to think about this on the whole body level. So if we account for the mass of each of these tissues and their glycolytic intermediate pool and sum them all up to measure a whole body glycolytic intermediate pool, what we find is that the majority of glycolytic intermediates come from glycogen, while only a small fraction come from circulating glucose. So glycogen is a major source of glycolytic intermediates. However, this is a little bit different than what I told you at the beginning of the talk, that glucose makes circulating lactate, and that the majority of circulating lactate actually comes from circulating glucose. So here's what I mean. So here on the left is the whole body measurement of the sources of glycolytic intermediates, where most glycolytic intermediate come from glycogen. And then on the right, in the same experiments, I can calculate the sources of circulating lactate. And the major source of circulating lactate is glucose. So these seem to be in contradiction. However, one thing can explain this seeming contradiction, and that is that we think glycolysis is compartmentalized. So what do I mean by that? So here on the left, I'm showing an example of if glycolysis was not compartmentalized. If glycolytic intermediates in the body were one single pool, then we would expect that the product of this pool, lactate, would have equal ratio of labeling from glucose to glycogen. But we know this is not true, because we know each tissue or organ has its own glycolytic intermediate pool. And therefore, tissues that use glucose, such as red muscle, may account for less of a fraction of the overall glycolytic intermediate pool, but it may have a higher rate of lactate production. While tissues that use glycogen for glycolysis may account for a large fraction of the glycolytic intermediate pool, but produce lactate at a slower rate. Therefore, when we look at whole body circulating lactate, we see much higher amounts of lactate labeling from circulating glucose. So this can be totally explained by compartmentalization of glycolysis. And we think, red muscle is an important organ for this. But we also think this may be happening on the cellular level, where heterogeneous tissues may have some cell types that are more glycolytic or use glucose for glycolysis. So the next question that I want to ask is glycolysis compartmentalized within individual cells or cell types? And we do have some evidence for this. So when we infuse glucose into the mouse, we can measure the labeling of lactate in the artery, and this represents whole body metabolic activity. But what we can also do is we can collect blood from a tissue draining vein, and then this will reflect the metabolic activity in the tissues that drain into that vein. And so here I'm showing data for the portal vein, and the portal vein drains the stomach, the intestines, the pancreas, and the spleen. And what we can see here is that the labeling we observe in the portal vein, lactate, is much higher than what we see in the artery. So this suggests that there is a tissue or cell in the portal bed that is important for, that does a lot of glycolysis from glucose. However, when we look at all of these tissues in the portal bed, we don't see a tissue that labels its glycolytic intermediates more than circulating lactate. So that suggests there's a cell type in one of these tissues that's doing a lot of glycolysis and producing lactate at a high rate, but it is a minority cell type in terms of it accounts for only a minority of the glycolytic intermediates in that tissue. So we think that glycolysis is also compartmentalized into specific cell types within tissues. And so, for example, the intestine would have a cell type that's using glucose and producing lactate at a high rate, while glycogen may be used in the rest of the cells and account for a lot of glycolytic intermediate, but not a lot of lactate production. So also, I won't have time to talk about it today, but our lab recently published a paper showing that this also may be true in tumors. So we observed differential labeling of glycolytic intermediates and lactate in tumors, suggesting that there's a highly glycolytic cell type producing lactate that is not represented in the bulk tumor glycolytic intermediate pool. So overall, we think glycolytic flux is concentrated in certain tissues and cell types in the body. So here on the top, I'm showing the whole body glycolytic intermediate pool. Most of it comes from glycogen, while only a small portion comes from glucose. However, when we look at the flux of lactate production, we see a large fraction of lactate comes from glucose, and therefore this small pool has a much higher flux out of it, while the breakdown of glycogen seems to produce lactate at a much slower rate. So in conclusion, we think glycolytic flux is compartmentalized. And what we're really interested in going forward is what cell types within heterogeneous tissues are glycolytic. And so all of this work really highlights some of the limitations of LC-MS, because we need enough material to be able to extract metabolites and measure on a mass spec. We usually grind up whole tissues and measure bulk labeling. But what would be really a lot better would be to be able to measure labeling in individual cells within that tissue. So to start moving towards this goal, we've been using MALDI imaging mass spectrometry. So this is in collaboration with Sean Davidson, who's a Louis Sigler fellow at Princeton, and another postdoc in the lab, Lin Wang. And MALDI stands for Matrix Associated Laser Desorption Ionization. So basically what this means is we take a tissue slice and we deposit a matrix on top of the tissue, and this matrix aids in ionizing metabolites within the tissue. So we can excite the tissue with a laser, ionize metabolites, and those metabolites can go into the mass spec and be measured just like traditional mass spectrometry. And because we can do this by shining a laser spot, we can measure the abundance across the tissue and generate an image of metabolite abundance within that tissue slice. And so you can see here is malate, and you can see there's a region in which malate is more abundant within this tissue. To look at glycolysis, we combine this with infusion of carbon-13 labeled glucose, take tissue sections, and then look for glycolytic intermediates that have labeling to determine which cells within that tissue are doing glycolysis. And because we're just starting using this methodology, I wanted to start with a tissue that has very dramatic differences in labeling from glucose. And so a tissue that we know from our LC-MS data has large differences in labeling are different types of muscle. And gastrocnemius and soleus muscle are right next to each other. So that means we can take these muscles out of the mouse and we can section them and we can view them within one section. So what you can see here is the soleus muscle that is labeling for a lot of type 1 and type 2A fibers. This is immunofluorescence. The plantaris muscle, which has some of these type 2A fibers, but is mostly type 2B. And then the gastrocnemius muscle, which is majority these type 2B fibers, which I discussed previously, make up white muscle because they don't have a lot of mitochondria or myoglobin. But the gastrocnemius muscle also has some regions that are a little bit redder, for lack of a better term, where we have more type 1 and type 2A fibers. So when we look at metabolite abundances across these muscles, what we can see is when we look at a glycolytic intermediate, fructose bisphosphate, that this glycolytic intermediate is very abundant in the wider regions of the muscle, or where there's a lot of type 2B fibers. And this is what is classically known about white muscle, is that it has high glycolytic capacity and has a lot of glycolytic intermediates and enzymes. And then when we look at malate, we can see that it's very high in the soleus and the redder regions of the gastrocnemius and the plantaris muscle. And this is also what is known, is that there's a lot of mitochondria in these fibers and therefore more TCA cycle intermediates. However, what we want to know is which muscle fibers are actively using glucose and glycolysis. So we looked at labeling in these intermediates. And so on top here is the data I showed on the last slide, but on the bottom is the labeled fraction of fructose bisphosphate, and this is the M+6, the fully labeled form. And what you can see is, while fructose bisphosphate is not abundant in the soleus, the labeled fraction is very high, suggesting it's actively catabolizing glucose. And so this matches very well with our LCMS data. But what is even more striking is that you can see the redder regions of the gastrocnemius muscle and the plantaris muscle are also labeling more than the wider regions, which seems to really confirm our conclusion that red muscle is very actively using glucose and glycolysis. And we can look at all of the glycolytic intermediates that I showed LCMS data for, and they all show the same pattern where redder muscles seem to use glucose directly. And so we think this is a really powerful tool, and we're moving forward and looking at more tissues to identify glycolytic cell types, and also in improving our resolution so that we can get closer to single cell resolution. So that is the end of part 1. So glycogen is a major source of glycolytic intermediates, while glucose is the major source of circulating lactate. So combined, this really suggests that glycolytic flux from circulating glucose is compartmentalized. And we think some major sites of glycolysis are red muscle, some cells within the intestine, and probably other cell types within other heterogeneous tissues. And I'm really interested in this difference in muscle metabolism. This importance of red muscle in using circulating glucose seems to align really well with the literature, where it's seen that the amount of oxidative fibers or red muscle is decreased in metabolic disease, such as obesity and diabetes. And exercise, which is known to improve glycemic control, both increases glucose uptake in white muscle and increases the amount of red muscle. So I think it's really interesting to think about strategies that could increase the amount of red muscle or increase glucose use in white muscle to treat type 2 diabetes. And I think that this is an idea that's been around for a while, but my data seems to support that this would be a really interesting avenue. And finally, I think it's interesting to rethink our view of carbohydrate metabolism. Classically, we think about almost all cells taking up glucose and then making the choice whether they want to use the pyruvate generated from glycolysis in the TCA cycle or for production of lactate. But based on our in vivo data, it seems that most tissues used lactate in their TCA cycle, but what seems to be optional is glycolysis, or at least the rate of use of glycolysis. So I think it's interesting to think about whether glycolysis itself is optional and whether tissues could survive in the absence of glycolysis. So in the second half of my talk, I would like to talk about de novo lipogenesis. And as I mentioned previously, lipogenesis is one fate of circulating glucose. However, when oxidation and glycogen storage is really saturated, this is when lipogenesis becomes a really good option for storing glucose. And so eating excessive carbohydrates or sweets really can drive the accumulation of fat in the liver, and excess carbohydrates that we intake in the form of carbs and protein can be converted into fat through the process of de novo lipogenesis. And the building blocks of de novo lipogenesis are really acetyl-CoA and NADPH. And this pathway, de novo lipogenesis, is known to be increased in patients with non-alcoholic fatty liver disease, thanks to some really nice work by Elizabeth Park's lab. So lipogenesis occurs through breaking down nutrients into acetyl-CoA, and then these acetyl-CoAs will provide the carbon to make fatty acid chains. However, to do this, you need reducing power. And so that reducing power comes in the form of NADPH, and that is required to produce fat. So we became interested in whether prevention of NADPH or inhibition of NADPH production could block de novo lipogenesis, and whether this could inhibit the buildup of fat in the liver. And so I started working on this project with another grad student in the lab, Jiaoye Zhang, who recently graduated and is now in Jeff Friedman's lab. So in order to block NADPH production in the liver, one thing we need to know is where that NADPH comes from. So our main question in this project is, what metabolic pathways produce NADPH in the liver? And there are several known pathways that can produce NADPH in cells. And one of the canonical pathways is the oxidative pentose phosphate pathway. And so because of the prevalence of this pathway in the literature, we decided to start with this. So the oxidative pentose phosphate pathway uses glucose to make NADPH. And so we can use stable isotopes to measure the activity of this pathway. So instead of using carbon-13 labeled glucose, we can use deuterated glucose, where the hydrogen at the position, on the first carbon that will be transferred to NADPH can be labeled. And therefore we can measure the labeled hydrogen in NADPH only if this pathway is active within a tissue. Furthermore, if this NADPH is used for lipogenesis, we should also measure labeling into fatty acids in the tissue. So we infused this tracer into mice for 12 hours overnight because lipogenesis is quite slow, and then looked at the labeling in tissues. And so what we saw was a little bit surprising. We saw almost no labeling in the liver, but we saw a lot of labeling in the brown adipose tissue. And so this suggests that OXPPP is not active in the liver. And when we looked at fatty acids, so here's palmitate, we saw the same thing. There was no labeling in the liver, but extensive labeling in the brown adipose tissue. So it seems that the oxidative pentose phosphate pathway, or OXPPP, supports lipogenesis in brown adipose, but not in the liver. So what pathway is producing NADPH? In looking at the other pathways, that can produce NADPH, we became very interested in serine catabolism. And that's because several of the enzymes involved in this pathway are actually highly expressed in the liver. So we used a similar strategy in which we used deuterium labeled serine, where the hydrogens that will eventually be used to make NADPH are labeled. This labeled serine will make a labeled 5-methylene THF, 5,10-methylene THF, and this will be used to generate NADPH. So when we infuse this overnight, just as we did with the glucose tracer, what we see is labeling in the liver, but no labeling in brown adipose tissue. So this suggests that serine catabolism is actually used for NADPH production in the liver. So is this used to drive lipogenesis? Yes, we can see labeling in the fatty acids in the liver also, and we could not see labeling in any other tissue. So, we concluded that serine catabolism produces NADPH for lipogenesis in the liver. However, we wanted to address our initial hypothesis: can we inhibit NADPH production and decrease de novo lipogenesis? So to do this, we used two tools to inhibit serine catabolism. One is a SHMT1 knockout mouse that lacks SHMT1, which converts serine to glycine and produces the 5,10-methylene THF that is used to produce NADPH. And our lab also has developed a SHMT deuterium, which we call SHIN2. So using both of these tools, we infused this deuterated serine to look at NADPH production, and both of these tools can block NADPH production through serine catabolism in the liver. So using these tools, we wanted to see if inhibiting serine catabolism decreases lipogenesis in the liver. So to measure lipogenesis, we used a classic tool, which is deuterium oxide, which will incorporate labeling only into newly synthesized fat. And so when we measured lipogenic flux, we found that when we inhibited serine catabolism, either by knocking out SHMT1 or with an inhibitor, that lipogenic flux decreased in the liver. But when we looked at brown adipose tissue, there was no significant difference. So this was great. Inhibition of serine catabolism decreases lipogenic flux in the liver. However, this is in the healthy liver. So we wanted to look at a condition where there was abnormal amounts of lipogenesis, and we know that high amounts of sugar can drive lipogenesis. So we fed mice 20% sucrose water for 4 weeks, and we could confirm that this increased lipogenic flux in the liver more than twofold. So using these mice that have been fed 20% sucrose water for 4 weeks, we treated the mice acutely for 36 hours with our SHMT inhibitor. And then at the end, we measured lipogenic flux with D2O. So what we saw was really nice. We saw an inhibition of lipogenic flux in the liver. And we were happy to see a decrease in the levels of triglycerides, both in the serum and the liver. So in circulation, we found a broad decrease in triglycerides. And then in the liver, we saw a more select amount of triglycerides that were decreased, but the triglycerides that were decreased were composed of saturated fatty acids, which are the main fatty acids produced by lipogenesis. So in summary, in part 2, serine catabolism produces NADPH for lipogenesis in the liver, while OXPPP from glucose is responsible for producing NADPH in brown adipose tissue. And then when we inhibit serine catabolism, we can actually decrease lipogenesis in the liver. And so we're really interested in the role of this pathway in the development of fatty liver disease. And just to put this in context, there's a lot of enzymes in the lipogenic pathway that are being targeted in clinical trials for the treatment of fatty liver disease. And so the serine catabolism pathway seems to be another alternative target. And so we're really interested in looking into the role of this pathway and whether this is a good target. So in conclusion, I think it's really important to understand these underlying metabolic pathways that are dysregulated in these chronic diseases. And so moving forward, I'm interested in understanding further the role of glycolysis in normal physiology. Is glycolysis really optional? And if so, in what tissues? Furthermore, I'm interested in finding the cell types that are most important for regulation of circulating glucose and glucose consumption. And finally, I'm really interested in studying the importance of red muscle in maintaining circulating glucose homeostasis. And with that, I would like to thank my lab and my postdoc advisor, Josh Rabinowitz, who has helped me a lot in thinking about metabolism in a more quantitative way, and Jiaoyu Zhang, who's worked with me very closely on the de novo lipogenesis project and really has pushed that project forward. And I'm happy to get a lot of feedback, and I look forward to lots of questions and comments on Twitter. So thank you.